Somewhere in a cold Siberian cave, buried under thousands of years of dust and sediment, a single bone fragment sat waiting. Not a skull, not a jaw, not even a full finger, just the tip of a pinky bone from a young girl who lived tens of thousands of years ago. That's it. That's the fossil that rewrote what we thought we knew about human evolution. If you're settling in to sleep tonight, that's exactly why I make these. So follow this show on Spotify, listen there when you can, and if you enjoy it, leave a 5-star review. The link's in the description. It genuinely helps the channel keep going. And tell me in the comments where you're listening from and what time it is right now. I love checking in the morning and seeing London at midnight, Sydney at sunrise, Chicago somewhere in between. All of you drifting off together without even knowing it. Now, think about how strange this next part really is. We know an enormous amount about human history. We've mapped skeletons of Homo erectus. We've reconstructed the faces of Neanderthals down to their eyebrow ridges. We've traced the journey of our own species, Homo sapiens, across continents using thousands of fossils, tools, and cave paintings. But there is one entire branch of the human family tree that we mostly know through genetics alone. Not through skulls, not through skeletons, through DNA extracted from fragments, so small you could lose them in a fingernail clipping. That branch is called the Denisovans, and here's the almost unbelievable truth. We still don't know what they look like. Not really. Not with confidence. We know they existed. We know their genes live on inside millions of people walking around today. But if you asked a scientist to draw you a Denisovan face, they'd have to guess because the evidence is that thin. This is the strange, almost detective story we're diving into tonight. A human lineage discovered not because someone found a body, but because someone found a genetic signal buried inside a bone chip so tiny it was almost thrown away. Scientists had assumed for decades that they understood the outline of the human family tree. Modern humans over here, Neanderthals over there, some older ancestors way back at the roots. Then Siberia had other plans. A remote cave, tucked into the Altai Mountains, would eventually reveal a genetic surprise so significant it forced researchers to add an entirely new branch to our family tree, one that didn't match anything we had ever excavated before. And the story doesn't stop with just discovering that they existed. Because as researchers dug deeper, they found evidence that Denisovans didn't stay in one place. Their genetic fingerprints turned up not just in Siberia, but scattered across Asia, in the genomes of people living today in places like Papua New Guinea, Australia, and the Philippines, thousands of miles from that cold mountain cave. Somehow this population moved. Somehow they thrived across environments as different as frozen tundra and tropical rainforest. And somehow they crossed paths, repeatedly, with our own ancestors and with Neanderthals, leaving behind evidence of interbreeding written directly into human DNA. So how do you build a picture of an entire vanished people using a pinky bone, a few teeth, and fragments of jaw? How do scientists reconstruct a lineage's appearance, its range, its relationships, when nearly everything they have to work with can fit inside the palm of your hand? That's the investigation we're walking through tonight. Not a simple timeline of who lived when, but a genuine mystery. How science reads a story out of near silence, how researchers turn a whisper of ancient genetic code into evidence of migration, interbreeding, adaptation, even survival on some of the harshest, highest terrain on Earth. It's a story built from fragments, but the implications reach every single one of us, because pieces of the Denisovans quite literally are still alive today, hidden inside human DNA, waiting to be understood. For a long time, the story of human evolution looked simple, almost like a ladder, one species climbing up, replacing the one below it, until you reached us at the top. Homo erectus gives way to something more advanced. That gives way to something smarter, until finally Homo sapiens shows up and the ladder ends. It made for a tidy story. It was also mostly wrong. Real human evolution looks less like a ladder and more like a river delta. Branches splitting off, wandering in different directions, sometimes drying up, sometimes reconnecting downstream. Populations separating for tens of thousands of years, adapting to their own environments, then meeting again and mixing their genes right back together. Untangling that kind of history is a lot harder than climbing a ladder. By the time researchers started seriously investigating Asia, they already knew the picture had to be more complicated than modern humans and Neanderthals alone. Neanderthals occupied Europe and parts of Western Asia. Homo sapiens was expanding out of Africa. But Asia is enormous. Mountains, deserts, tropical islands, frozen plateaus. It seemed almost impossible that only two human groups had filled all of that space for hundreds of thousands of years. Something else was probably out there. The problem was finding it. Before ancient DNA technology existed, everything depended on physical remains. Bones, teeth, stone tools scattered through cave sediment. And Asia's fossil record from this period was thin, fragmented, and difficult to interpret. A tooth here, a jaw fragment there, nothing complete enough to confidently name a new species. So the gaps stayed gaps. Scientists suspected there was more to the story, but suspicion isn't evidence. And that left room for something remarkable to stay hidden in plain sight. An entire population of ancient humans waiting in the dark, leaving barely a trace until a single cave in Siberia forced everyone to look again. At the edge of southern Siberia, where the Altai Mountains rise between the great grasslands of Mongolia, the forests of northern Asia, and the high country leading toward China and Kazakhstan, there is a cave overlooking the Anui River Valley. It is called Denisova Cave. From the outside, it doesn't look like the kind of place that should rewrite the human family tree. It is not enormous. It is not decorated with famous paintings. There are no complete skeletons laid out in museum cases, no dramatic monuments announcing that something extraordinary happened there. Just a rocky opening in a limestone hillside above a river, in a region where winters are long, cold, and unforgiving. But location matters. Denisova Cave sits at a natural crossroads. To the north and west, open steppe stretches toward Kazakhstan and the vast interior of Eurasia. To the south, mountain routes lead toward Mongolia, China, and the Tibetan Plateau. Nearby forests offered wood, shelter, and game. The river valley created a passage through rugged terrain. For animals moving between ecosystems and for humans following those animals, this was not the end of the world. It was a meeting point. Picture the landscape during the Ice Age. The exact climate changed again and again, sometimes becoming colder and drier, sometimes allowing forests and grasslands to expand. Herds of bison, deer, ibex, horse, and other animals moved through the valleys. Predators followed: wolves, cave hyenas, lions, and humans, carrying stone tools, fire, food, and whatever knowledge had kept their families alive through another hard season. A cave like Denisova offered something rare: shelter from wind, protection from snow, a dry place to make tools, butcher animals, rest, and perhaps wait out the worst weather. Not necessarily a permanent home. More likely, across thousands of generations, a place people return to. One group leaves. Sediment slowly blows in. Animals occupy the space. Another group arrives years, centuries, or millennia later. They leave behind a few flakes of stone, broken bones from meals, charcoal from fires, perhaps a lost tooth or a fragment of fingerbone. Then they disappear into the larger landscape. The cave keeps the record. That is the strange power of caves. Open-air camps are often erased by rain, wind, roots, rivers, and later human activity. But inside a sheltered cave, layer after layer can survive. Dust settles, roof fragments fall, ash accumulates. Animal bones are dragged in. Human visitors leave traces so small that they may not look important at first. Over time, these materials become stacked like pages in a book. At Denisova Cave, that book is unusually thick. Archaeologists found sediments representing a very long span of time, reaching back hundreds of thousands of years. The dates are still refined as new methods improve, and not every layer is equally clear. But the general picture is remarkable. The cave was used repeatedly across much of the Middle and Late Paleolithic, through periods when climates shifted dramatically and different human populations moved across Eurasia. That alone made it important, long before anyone suspected that the cave held evidence of an unknown human lineage. Researchers recognized it as a valuable archaeological site. Soviet and Russian archaeologists began systematic excavations there in the late 20th century. They worked slowly because archaeology in a cave is less like digging a hole and more like reading a fragile archive one grain at a time. Every layer had to be recorded. Where was the sediment found? What colour was it? Was it compacted, sandy, rocky, full of ash, or disturbed by burrowing animals? Which stone tools appeared in the same level? Which animal bones? Which tiny fragments might belong to humans? This matters because a cave floor is not a single moment frozen in time. It is a mixture of countless moments. A bone can be moved by an animal. Water can shift sediment. A later visitor can dig into an older layer without knowing it. If archaeologists want to understand who was in the cave, and when, they need context. A fragment of bone by itself is interesting. A fragment of bone precisely dated and connected to a specific layer, tool tradition, climate phase and animal community becomes evidence. So the excavators sifted through sediment carefully. They collected stone flakes, blades, scrapers and points. They examined animal remains for cut marks, burning and signs of hunting or scavenging. They found objects made from bone and teeth. They studied pollen, microscopic particles, and the chemistry of the layers themselves. Sometimes the most valuable clues were not the biggest ones. A tiny piece of charcoal can help date a fire. A grain of sediment can preserve ancient DNA. A single tooth can contain a whole evolutionary history. And Denisova Cave had many stories layered on top of each other. Some of its stone tools suggested very ancient forms of technology, made by people who lived long before modern humans entered Siberia. Other layers contained more refined artifacts associated with the transition toward the Upper Paleolithic, a period when tools, ornaments, and symbolic objects become more common in parts of Eurasia. Animal bones told their own story of changing climates and environments. The species found in one layer were not always the same as those found in another. As the world cooled or warmed, the valley changed and the visitors to the cave changed with it. This is one reason Denisova Cave became so important. It was not simply a site with one occupation and one group of people. It was a crossroads used repeatedly over an immense stretch of time. Different human populations may have passed through the same chambers, sat near the same entrance, worked stone from the same local materials, and watched the same river valley from the same slope. They may have been separated by thousands of years. Or at certain moments they may have lived surprisingly close to one another. That possibility was already built into the geography. The Altai region lies far from the places most people associate with ancient human origins. It is not Africa, where Homo sapiens emerged. It is not Western Europe, where Neanderthal caves became famous. It is deep in Asia, in territory that had often been treated as a blank space on the map of human evolution. But blank spaces on maps are dangerous. They can make us confuse lack of evidence with absence. Just because few fossils had been found in a region did not mean few humans had lived there. It could mean the fossils were buried. It could mean they had not survived. Or it could mean that the Reich cave had not yet been examined closely enough. Denisova Cave was the Reich cave, not because it immediately gave up a spectacular skull or a nearly complete skeleton. In fact, what made it famous was almost the opposite. The decisive evidence would come from something so small that it could have been overlooked, misplaced, or dismissed as unimportant. A fragment from an ancient human body, resting quietly among the sediments of a Siberian cave. Before that fragment changed everything, Denisova Cave was already a place of deep time. A shelter used through changing ice ages. A record of tools, animals, fires, and vanished visitors. A crossroads between mountain, forest, and steppe. And then, in the layers of that old cave, archaeologists found a clue that did not fit the existing family tree at all. In 2008, that clue appeared, not as a skull, not as a dramatic skeleton, with a face you could reconstruct and place in a museum. It was a tiny piece of bone found in a layer of Denisova Cave known as Layer 11. The fragment came from the end of a finger, probably the little finger, and it was so small that it could rest easily in the palm of your hand. At first, there was no obvious reason to think it belonged to a previously unknown branch of humanity. Ancient human bones are often incomplete. They are broken by time, crushed by sediment, moved by water, chewed by animals, or scattered during excavation, and a finger bone is especially difficult to identify. A skull can show the broad brow ridge of a Neanderthal, or the rounded shape more typical of Homo sapiens. A jaw can preserve distinctive teeth. A thigh bone can reveal body proportions and walking style. But the tip of a finger tells you almost nothing by itself. The bone was small and gracile. It may have belonged to a young girl, perhaps around 5 to 7 years old, though estimates like that always carry uncertainty. Her name, if she had one, is gone. Her face is gone. Her body is gone. All that remained was one fragment of pinkish bone, less than 3 centimeters long. If this had been discovered a few decades earlier, it might have been catalogued as an unidentified ancient human fragment. Possibly Neanderthal, possibly modern human, maybe too incomplete to say much at all, and then it would have stayed in a drawer. But this was the 21st century. Archaeologists no longer had to rely only on the visible shape of bone. They could ask a more intimate question, not what does this fragment look like, but what genetic history is still locked inside it. Ancient DNA research is difficult, almost absurdly difficult. DNA begins breaking apart after death. Heat, water, oxygen, bacteria, and time all destroy it. Siberian caves, with their cold temperatures, can preserve genetic material better than many places, but better does not mean easy. The DNA in an ancient bone is usually fragmented into tiny damaged pieces. It is mixed with the DNA of microbes, animals, excavators, and everyone who has touched the specimen since it was uncovered. Getting a usable sequence is like trying to reconstruct an ancient book after it has been soaked, burned, torn into millions of scraps, and mixed with the pages of thousands of other books. Still, the researchers tried. The first genetic evidence came from mitochondrial DNA. Mitochondria are tiny structures inside almost every cell, often described as the cell's energy factories. They have their own small genome, separate from the much larger genome stored in the cell nucleus. Mitochondrial DNA is inherited almost entirely through the maternal line. Your mitochondria come from your mother, who got hers from her mother, and so on. back through a chain of mothers stretching into deep time, because each cell contains many mitochondria. Mitochondrial DNA is often easier to recover from ancient remains than nuclear DNA. It does not give the whole family story, it follows only one narrow ancestral thread. But sometimes one thread is enough to show that something strange is happening. When the mitochondrial DNA from the finger bone was analysed, the result was startling. It did not match modern humans. That alone would not have been shocking. By the time of the discovery, scientists already knew that Neanderthals were genetically distinct from living people, even though many people outside Africa carry some Neanderthal ancestry today. Denisova Cave was in Asia, and Neanderthals had lived across large parts of Eurasia. A Neanderthal fragment would have been important, but not unimaginable. But it did not match Neanderthals either. The mitochondrial lineage seemed to have split from the maternal line shared by Neanderthals and modern humans much earlier than expected. Early estimates suggested a separation reaching back around 1 million years. This did not mean the finger bone itself was 1 million years old. It was much younger than that. It meant that the maternal ancestry preserved in its mitochondria had been separate for an unexpectedly long time. For a moment, the old map of human evolution broke open. Was this an entirely unknown human species? Was it a surviving population descended from an ancient migration out of Africa? Was the mitochondrial result misleading, preserving an unusually old maternal line that had passed through a population otherwise related to Neanderthals? Mitochondrial DNA can reveal a surprise, but it cannot settle every question. A single maternal lineage is not the same thing as a whole population. The small finger bone had given science a mystery. To solve it, researchers needed the nuclear genome. This was the real test. Nuclear DNA contains the overwhelming majority of an organism's genetic information. It comes from both parents and carries evidence from ancestors across the family tree, not just one maternal line. If enough of it could be recovered from the fragment, scientists could compare this individual with Neanderthals, modern humans, and people living today. That was a huge technical challenge at the time. Recovering a complete ancient nuclear genome from such a tiny specimen was near the edge of what was possible. The bone had to be sampled carefully. The DNA had to be extracted in ultra-clean laboratories designed to reduce contamination. Sequencing machines had to read millions upon millions of microscopic fragments. Then computers had to sort ancient human DNA from everything else. And when the results came back, they revealed something even more interesting than the mitochondrial DNA had suggested. The girl from Denisova Cave was closely related to Neanderthals. Not a Neanderthal exactly, but their close evolutionary cousin. Modern humans, Neanderthals, and this newly recognized population shared a deeper common ancestry. Then, after the lineage leading to modern humans had separated, another split occurred. One branch led toward Neanderthals, mostly known from Europe and Western Asia. The other led toward the people now called Denisovans, whose genetic traces would eventually be found across Asia and Oceania. Think of it like 3 branches growing from one older trunk. Modern humans take one path, Neanderthals take another. And beside the Neanderthal branch, almost hidden in the darkness of Eurasia, was a sister branch nobody had recognized before. The finger bone was not simply from an unusual Neanderthal. It belonged to a distinct population with its own evolutionary history. That distinction matters. Scientists are careful with labels like species. The fossil record for Denisovans is sparse, and they clearly interbred with Neanderthals and modern humans. Whether they should be treated as a separate species, a subspecies, or a deeply distinct human population depends partly on definitions. Human evolution does not always fit neatly into the boxes we like to draw. But whatever label is used, the discovery was extraordinary. A whole branch of recent human history had emerged from a fragment of a little finger. Until then, the story of Eurasian humans had often been told as a two-character drama. Modern humans and Neanderthals, us and them, Africa and Europe. Then this tiny bone from southern Siberia revealed that the drama had another major character, one that had lived across vast regions of Asia and had left almost no obvious fossil trail behind. Not invisible, just overlooked, and the more scientists examined the Denisovan genome, the stranger the story became. This was not merely a lost population preserved in one Siberian cave. Their DNA was still alive, scattered in the genomes of people living thousands of miles away, from the high mountains of Tibet to the islands of the Pacific. The little finger had opened a door. Beyond it was a human world far more crowded, connected, and complicated than anyone had expected. But opening that door was not as simple as placing the finger bone into a machine and waiting for an answer. Ancient DNA is not preserved like a message in a bottle. It is more like smoke after a fire. Most of it is gone. What remains has been broken into tiny pieces, altered by chemical reactions, mixed with DNA from bacteria, fungi, cave animals, plants, archaeologists, museum workers, and modern humans, who may have handled the specimen at any point in the last several thousand years. The challenge was not just finding DNA. The challenge was proving that the DNA belonged to the ancient girl. Researchers begin with extreme caution. The outer surface of an old bone is often the most contaminated part. It may have been touched during excavation, washed, measured, photographed, or stored in conditions where microscopic modern DNA could settle onto it. So scientists remove or clean the surface, then take a small sample from the protected interior. Sometimes they drill into the bone, sometimes they powder a tiny portion of it. The amount can be very small, but even a small sample contains millions of cells, and inside some of those cells may be fragments of the original person's DNA. Then comes the extraction. The powdered bone is treated with chemicals that dissolve mineral material and release whatever DNA molecules remain. But the resulting liquid is not a neat human genetic sample. Usually the overwhelming majority of DNA comes from microbes that colonize the bone after death. Ancient human DNA may be only a tiny fraction of the total. Imagine searching through the dust of an abandoned library for scraps from one particular book, while nearly every scrap belongs to something else. This is where modern sequencing changed the field. Earlier genetic methods often needed scientists to target one specific stretch of DNA and copy it again and again. That worked for some questions, but ancient DNA is too damaged and too scarce for many traditional techniques. Newer sequencing methods can read enormous numbers of fragments at once. Millions, sometimes billions, of tiny genetic pieces can be collected and compared with reference genomes from modern humans, Neanderthals, animals and microbes. A computer then begins the slow sorting process. This fragment looks human. This one looks bacterial. This one may come from a cave bear. This one matches modern human DNA almost perfectly and may be contamination. This one is short, damaged, and genetically unlike any living person. That one may be ancient. It sounds almost magical. But the magic is really statistics, chemistry, and patience. Ancient DNA research depends on patterns. One isolated genetic fragment means very little. But thousands or millions of fragments, all showing the same tendencies, can reveal a reliable signal. And one of the most important signals is damage. DNA does not simply break apart with age. It changes in recognizable ways after death. Water and chemical reactions alter certain DNA bases. One common form of damage causes cytosine, one of the letters in the genetic code, to be read as thymine. This happens especially near the ends of ancient DNA fragments, where the molecules have been exposed and decaying for thousands of years. So when scientists sequence DNA from a genuinely old bone, they often find short fragments with a distinctive pattern of changes at their edges. That pattern is like a chemical fingerprint of time. It does not tell researchers the exact age of a specimen by itself. DNA damage depends on temperature, moisture, burial conditions, and countless other factors. A bone in a warm, wet environment can decay much faster than one in a cold, dry cave. But damage patterns help answer a critical question. Is this DNA likely to have come from an ancient individual, or did it come from a modern person who touched the bone? Modern contamination tends to be longer, less chemically damaged, and genetically closer to the people handling the sample. Ancient DNA tends to be short, degraded, and marked by those characteristic changes. Researchers also estimate contamination by looking at genetic positions where ancient populations and modern humans differ. If a sample contains too many modern-looking sequences, especially from the researchers themselves, the results become uncertain. This is why ancient DNA laboratories are so strict. The work is done in specially controlled spaces. Air is filtered. Surfaces are cleaned. Tools are sterilized. Researchers wear protective clothing, masks, gloves, and coverings that reduce the chance of shedding their own skin cells into the sample. In many cases, ancient DNA work is physically separated from laboratories where modern DNA is handled. Even then, no one assumes contamination has been eliminated completely. The goal is to measure it, understand it, and make sure it cannot explain the result. Because the Denisova finger bone carried such a small amount of usable human DNA, every fragment mattered. The first genetic clue had come from mitochondrial DNA, and there was a good reason for that. Mitochondria are the tiny structures inside cells that help produce energy. Each mitochondrion has its own small circle of DNA, and because cells contain many mitochondria, but only one nucleus, mitochondrial DNA can survive in greater numbers than nuclear DNA. If nuclear DNA is the complete family archive, mitochondrial DNA is one thin notebook copied down through mothers. You inherit it from your mother. She inherited it from her mother. Go backward long enough, and you can trace an unbroken maternal line into the past. This makes mitochondrial DNA useful for identifying broad relationships and surprising divergences. It was mitochondrial DNA that first showed the Denisova bone did not fit comfortably among known modern humans or Neanderthals. But mitochondrial DNA has limits. It follows one ancestral path out of thousands. Your parents each have two parents, those four grandparents each have two parents. The number of ancestors expands rapidly as you move back through time, though family lines overlap in real populations. Mitochondrial DNA ignores almost all of that complexity. It records only the chain of mothers. A population can acquire a different mitochondrial lineage through interbreeding. It can lose one through chance. It can preserve an old maternal branch long after the rest of its ancestry has changed. So mitochondrial DNA can announce that something unusual is happening. But it cannot by itself tell the whole story. Nuclear DNA can. The nucleus contains almost all of the genome spread across chromosomes inherited from both parents. It carries evidence from countless ancestors. It can reveal how closely two populations are related when they may have diverged, whether they exchanged genes after separating, and which stretches of DNA were later passed into living people. Recovering nuclear DNA from ancient remains is much harder because there is less of it per cell. But the Denisova sample came from an unusually favourable place. Denisova Cave lies in the Altai Mountains of southern Siberia, where the climate can be harsh and cold. For ancient DNA, cold is a gift. Low temperatures slow many of the chemical and biological processes that destroy genetic material. A cave can also provide a relatively stable environment, protecting bones from repeated cycles of heat, rain, sunlight, and surface disturbance. Not every layer of a cave is equally protective. Not every bone from Denisova Cave preserves DNA. Time still wins eventually. But compared with a warm tropical forest, an open grassland, or a damp coastal site, a cold Siberian cave can act like a kind of imperfect natural freezer. That is one reason Denisova Cave became so important. It did not merely preserve bones, it preserved molecular traces of people whose anatomy had almost vanished. When researchers compared the finger bone's nuclear genome with genomes from Neanderthals and living humans, the result was clear. The individual was neither a modern human nor a Neanderthal. Yet she was genetically closer to Neanderthals than either group was to us. This is how kinship can be reconstructed even when fossils are scarce. Scientists compared genomes letter by letter across millions of positions. If two populations share many genetic changes that are absent in a third, that suggests they inherited those changes from a more recent common ancestor. The Denisovan and Neanderthal genomes shared patterns indicating that their lineages had remained together after the lineage leading to modern humans had already gone its separate way. Picture three relatives trying to reconstruct a family history from old letters. One person shares certain phrases with another. The second and third share a different set of phrases. By comparing enough letters, you can work out who is most closely related, even if the family portraits are missing. The genome did not give researchers a face. It could not tell them exactly what Denisovans looked like, what language they spoke, or how they understood their world. DNA is powerful, but it is not a time machine. It gives probabilities, relationships, and biological clues. It cannot restore every lost detail, but it could reveal something extraordinary. The ancient girl from the finger bone belonged to a population that had separated from the Neanderthal line long before either group encountered modern humans in the later chapters of prehistory. They were close relatives, but not the same people. Two branches from a shared Eurasian ancestry, each following its own path through a changing continent. And now that the genome had spoken, the next question became unavoidable. If Denisovans were the Neanderthals' sister lineage, when had the two branches separated, and what kind of human world had existed before that split? The answer begins with a family tree, but not the simple kind, with clean branches and neat labels. A better picture is a river system, long ago, perhaps somewhere between 550,000 and 765,000 years ago, depending on the genetic method used and the assumptions built into the calculation. The ancestors of modern humans separated from the ancestors of a population that would later give rise to Neanderthals and Denisovans. One branch remained primarily in Africa. Over a long and complicated history, it led to Homo sapiens. The other branch moved through Eurasia, or perhaps was already spread between Africa and Eurasia in populations. We can no longer identify clearly from fossils. This ancestral Eurasian population was not yet Neanderthal. It was not yet Denisovan. It was a shared source population carrying the ancestry of both. Then, hundreds of thousands of years later, that Eurasian branch divided again. One line became the Neanderthals. The other became the Denisovans. That is what scientists mean when they call Denisovans the sister lineage to Neanderthals. Not sisters in the everyday sense, of course. Not two individuals standing side by side in a cave. Sister lineages are branches that share a more recent common ancestor with each other than either does with a third branch. Modern humans, Neanderthals, and Denisovans were close relatives. But in the deepest structure of the genetic evidence, Neanderthals and Denisovans were the closer pair. Picture three cousins whose family records have mostly been destroyed. You have only scraps, a few old photographs, some letters. Perhaps a box of objects passed down through generations. Modern humans share one set of inherited features. Neanderthals and Denisovans share another. Those shared features do not mean they were identical. They mean that, for a while longer, their ancestors belonged to the same population after our own ancestors had already begun following a separate path. Eventually, that shared population split too. Genetic estimates often place the separation between the Neanderthal and Denisovan lineages somewhere around 380,000 to 470,000 years ago. But those numbers are not dates stamped onto a calendar. They are estimates, and estimates in deep prehistory come with wide margins of uncertainty. Scientists calculate them by measuring genetic differences between genomes, then using models of mutation rates, generation lengths, population sizes, and later interbreeding. Change any one of those assumptions and the date can move. If mutations accumulated slightly faster than expected, the split may have happened more recently. If ancient populations had long generations, or if they continued exchanging genes after beginning to separate, the apparent date can shift. New genomes can change the picture too. A single individual from an unexpected place carrying ancestry from an unknown population can force researchers to redraw parts of the tree. And this is one of the most important things to remember about ancient DNA. A divergence date does not mean one afternoon, one valley, one couple, or one dramatic moment when a new species suddenly appeared. Evolution almost never works like that. Populations separate gradually. Groups move apart. Mountains, deserts, glaciers, forests and distances reduce contact. Over generations, people in one region tend to have children with people nearby. Local traits become more common. Genetic differences slowly accumulate. Then climates change. Rivers open new routes. A few groups meet again. They exchange genes. They separate once more. The real story is less like a tree splitting cleanly in two. It is more like a braided river. Channels divide, rejoin, disappear underground, and emerge again miles away. This matters because the word Denisovan can sound more precise than the evidence really allows. It sounds like a single uniform people, one clearly defined species, one population with a single appearance, a single culture, and a single territory. But that is almost certainly not what Denisovans were. The Denisovans, known from genetic evidence, had an enormous geographic reach. Their DNA later appeared in modern populations from East and Southeast Asia to New Guinea and the Western Pacific. Some of their ancestors, or closely related groups, lived in Siberia. Others contributed genes to people whose descendants would live on tropical islands. Still others seem connected to the high-altitude populations of the Tibetan Plateau. No population can occupy environments that differ for hundreds of thousands of years and remain perfectly uniform. There were probably Denisovan populations, plural. Some may have lived in cold northern landscapes. Some may have adapted to forests, grasslands, mountains, coastlines, and island chains. They may have been separated for long periods, developing their own genetic patterns and local ways of life. Some groups may have disappeared without leaving fossils. Others may survive only as faint stretches of DNA in living people. So Denisovan is useful. Scientists need names. Names let us discuss patterns, compare samples, and organize evidence. But every label is also a simplification. The same is true of Neanderthal and modern human. These names can make ancient populations seem like three separate boxes, each sealed shut. In reality, the boxes had openings. Neanderthals and Denisovans exchanged genes with each other. Both later exchanged genes with modern humans. Genetic evidence also hints at contact with even older populations, sometimes called ghost populations because we can detect their contribution in DNA without having a fossil or a name for them. There may have been communities where these groups met repeatedly over thousands of years. Communities where people who looked somewhat different still recognized each other as possible partners, allies, rivals, or neighbors. Communities where ancestry crossed boundaries that modern science is still trying to define. This does not mean the labels are meaningless. Neanderthals had their own long evolutionary history, especially in Europe and Western Asia. Denisovans had another, apparently centered farther east across Asia, though the precise limits remain uncertain. Modern humans developed primarily in Africa before expanding outward. Those broad patterns are real, but the boundaries were not walls. They were zones of contact, and the farther back we go, the harder it becomes to say exactly where one population ended and another began. Imagine standing on a wide open plain at dawn. Nearby shapes are clear. A person, a tree, a boulder. Farther away, everything begins to blur into the mist. You can still see that something is there. You can trace broad outlines. But you cannot confidently describe every edge. That is Eurasian human diversity before the arrival of modern humans. For hundreds of thousands of years, Eurasia was not empty, waiting for our species to enter and take over. It was occupied by multiple human populations, each adapting to local climates and landscapes, each carrying histories far older than any written record. Neanderthals were one part of that world, Denisovans were another, and behind both may have stood older populations whose names, bones and stories have been mostly erased. The finger bone from Denisova Cave gave this lost branch a name. The genome showed its nearest known relationship, but it also revealed the scale of what remained unknown. If Denisovans were a sister lineage to Neanderthals, where were their bodies? What did they look like? What could a handful of teeth, bone fragments, and one astonishingly ancient jaw tell us about people whose genetic legacy stretched across a continent? And that question brings us to one of the strangest facts in all of human evolution. For a population that contributed DNA to millions of people alive today, Denisovans have left remarkably little of themselves behind. Not cities, not cemeteries, not dozens of nearly complete skeletons lined up in museum drawers. Mostly they have left fragments, a small finger bone, a few teeth, pieces of long bone, tiny scraps of skull, a jawbone from far away on the Tibetan Plateau, and hidden within caves and living genomes, a genetic trail far larger than the fossil trail. At Denisova Cave itself, the human remains are so limited that they can be listed almost one by one. There was the little finger bone that started the story. It came from a young girl, now known as Denisova 3. Her genome revealed that she belonged to a population distinct from both modern humans and Neanderthals. There were teeth, several of them. There were fragments of bone from individuals whose bodies are otherwise gone. A toe bone that turned out to belong to a Neanderthal woman. A small piece of long bone from Denisova 11, the girl whose mother was Neanderthal and whose father was Denisovan. Other bits of bone and tooth have yielded DNA or proteins that connect them to the Denisovan side of the ancient Eurasian family. It is an extraordinary collection scientifically, but physically it is almost nothing. Imagine trying to reconstruct an entire family from a child's finger, a few loose molars, and pieces of bone no larger than a hand. You might be able to tell that these people existed. You might identify some biological relationships. You might even recover much of their genetic history, but you could not confidently picture their faces. And yet the teeth do give us something. The Denisovan molars from Denisova Cave are unusually large, not merely a little larger than average modern human molars, but strikingly broad and robust. Their crowns have complex shapes, and some possess features more often seen in much older human populations than in recent Homo sapiens. One upper molar, often called Denisova 4, is especially impressive. It is large enough that when scientists first examined it, it did not fit comfortably into the expected range for Neanderthals or modern humans. Another molar associated with a Denisovan individual also appears unusually large and distinctive. Teeth are among the toughest parts of a human body. They survive burial better than delicate facial bones. They preserve wear patterns, enamel structure, and tiny details of shape. For paleoanthropologists, they can be like durable little identity cards. But identity cards have limits. Large molars do not automatically mean every Denisovan had giant teeth. A few individuals cannot tell us the full range of variation in an entire population spread across Asia for hundreds of thousands of years. Some traits may reflect age, sex, local ancestry, diet, or simple individual variation, and even if large molars were common among some Denisovans, teeth cannot tell us everything else we want to know. They cannot tell us the exact shape of the nose. They cannot tell us whether Denisovans had heavy brow ridges like many Neanderthals, or a face that looked more like another ancient Asian human population. They cannot tell us the texture of their hair, the shade of their skin, the sound of their voices, or whether people in one Denisovan population looked much different from people thousands of miles away. For a long time that was almost the entire physical picture. Then, far to the south of Siberia, on the edge of the Tibetan Plateau, another fragment changed the story. In 1980, a Buddhist monk found a massive lower jaw in Baixia Karst Cave, near the village of Jiahe in China's Gansu province. The cave sits high above sea level, on the northeastern margin of the Tibetan Plateau, in a landscape where thin air makes ordinary physical work difficult for people who are not adapted to altitude. The jaw was ancient. It was large. It had no DNA that scientists could recover directly from the bone at the time. And without DNA, assigning it to a particular human group was difficult. For decades it remained a mystery. Then researchers used another method. Proteins' DNA is fragile. Over long periods, heat, water, microbes, and chemical change break it apart. Proteins also degrade, but some can persist where DNA does not. In the Jiahe jaw, scientists recovered ancient proteins from a molar. By comparing their amino acid sequences with those of modern humans, Neanderthals, and the Denisovan genome from Siberia, they found that the jaw belonged to a population closely related to Denisovans. Not a perfect label stamped into the bone, but strong evidence. The jaw, dated to at least around 160,000 years ago, showed that Denisovan-related people had reached the Tibetan Plateau long before modern humans arrived in the region. Later, genetic evidence from sediments inside Baixia Karst Cave strengthened the connection. Ancient DNA recovered from cave deposits included Denisovan mitochondrial DNA, showing that Denisovan-related populations had used the cave repeatedly across long stretches of time. The cave did not preserve a full Denisovan skeleton, but it preserved their presence, and the jaw itself added a few more clues. Its body is thick and powerful, its molars are large. Unlike modern human lower jaws, it lacks a projecting chin. In those broad features it resembles some earlier archaic human fossils from Asia. But again, resemblance is not certainty. A jaw can tell us about jaws. It cannot provide a complete portrait. This is where the popular image of Denisovans begins to outrun the evidence. You may see paintings of them with enormous brows, broad noses, dark hair, muscular bodies, and heavy, ancient-looking faces. Some portraits make them look almost like Neanderthals moved east. Others make them look like a blend of Neanderthal, modern human, and something unknown. These images are understandable. Humans want a face. we find it hard to imagine a relative who exists only as a genome and a tooth. But most Denisovan portraits are, at best, informed speculation. Scientists can make limited evidence-based inferences. Denisovans were human relatives close enough to Neanderthals and modern humans to have children with both. Some probably had robust jaws and large molars. At least some populations lived in cold Siberia. Others lived at high elevation on the Tibetan Plateau. Their genes later helped some modern Tibetan populations cope with low oxygen. Those are real clues, but they do not add up to a reliable, full-body reconstruction. Even attempts to infer anatomy from DNA remain uncertain. Researchers can sometimes examine ancient genetic variants associated in living people with aspects of skeletal development or physiology. Such studies may suggest possible tendencies, but genes interact with one another, and bodies are shaped by diet, childhood health, activity, climate, and development. A prediction from a genome is not the same thing as a skull recovered from the ground. The honest Denisovan face is still partly blank, not because science has failed, because science is refusing to pretend it knows more than it does. A few teeth tell us there were distinctive mouths. A finger bone tells us of a young girl in a Siberian cave. A huge jaw tells us that Denisovan-related people endured the heights of Tibet. Protein and DNA tell us these fragments belong to a branch of humanity we never knew existed. But the rest remains in shadow, and that shadow grows larger when we leave Denisova Cave behind. Because if Denisovan DNA is found today from the Himalayas to Ireland, Southeast Asia and the Pacific, then the people represented by these few Siberian fragments cannot have been the whole story. Somewhere across Asia there must be more of their history waiting in caves, river valleys, mountain shelters and tropical landscapes where bones vanish quickly. The challenge is knowing where to look. The first answer came from Tibet. Not a complete answer, not even close, but the Jahe jaw made one thing impossible to ignore. Denisovan history was never just a story about one cold cave in the Altai Mountains. Denisova Cave is in southern Siberia, thousands of miles from the Tibetan Plateau. Between them lie deserts, forests, river systems, mountain ranges, grasslands, and some of the most difficult terrain on Earth. A Denisovan-related jaw high on the edge of Tibet meant that these ancient people or closely related populations had moved far beyond the place that gave them their name. And once scientists began looking at living DNA rather than fossils alone, the map expanded even further. Modern people in Tibet carry a remarkable genetic variant called EPAS1. It helps regulate the body's response to low oxygen. At high altitude, where every breath contains less oxygen than it does at sea level, this kind of adaptation can make the difference between living comfortably, becoming dangerously ill, or never being able to establish a population there at all. The Tibetan version of EPAS1 is unusual. It is rare or absent in most other modern human populations. But it closely resembles a version found in the Denisovan genome from Siberia. The most likely explanation is that the ancestors of Tibetans inherited it through interbreeding with Denisovan-related people. Later, once modern humans were living on the plateau, natural selection strongly favoured the variant. People carrying it were better able to cope with thin air. They were more likely to survive, have children, and pass it on. So a gene from an ancient population became part of a modern, high-altitude way of life. That does not necessarily mean the Denisovans of Siberia themselves marched across Asia and settled every corner of Tibet. The genetic picture may be more complicated than that. The people at Jahe, for example, may have belonged to a population related to the Siberian Denisovans, but not identical to them. They may have been separated for tens of thousands of years, developing their own local history, their own appearance and their own adaptations. Think of Denisovans less as one tiny group trapped in a cave, and more as a wide human branch with regional populations spread across enormous distances. That is what the DNA increasingly suggests. The strongest Denisovan ancestry in living people is found far to the southeast, especially among populations in Melanesia, including people of Papua New Guinea and nearby islands. In some of these populations, several % of the genome comes from Denisovan-related ancestors. That may not sound like much, but across an entire human genome, it represents millions of inherited genetic letters passed down through countless generations. It is a much larger signal than anyone would expect if Denisovans had lived only in Siberia, and it raises an obvious question. How did a population known from a cave in the Altai Mountains leave such a clear genetic legacy in people living near New Guinea? One possibility is that Denisovan-related groups were once widespread across mainland Asia. As modern humans moved east after leaving Africa, they may have encountered different Denisovan populations in different regions. Some encounters may have happened in mainland Southeast Asia. Others may have occurred farther east, perhaps near the ancient land routes that once connected islands now separated by sea. During the Ice Ages, sea level was much lower than it is today. Vast areas of shallow ocean were dry land. Mainland Southeast Asia, Sumatra, Java, Borneo and other islands formed a larger connected region often called Sunda. It was not one simple open plain. Rivers crossed it. Forests covered parts of it. Mountains rose in the distance. But routes that are now underwater could once have allowed humans and animals to move across a much broader landscape. Beyond Sunda lay Wallacea, the island zone leading toward New Guinea and Australia. Even when sea levels were low, some water crossings remained. Reaching the far eastern islands required boats, rafts, or some other way of deliberately crossing open water. Modern humans certainly managed that journey by at least around 50,000 years ago, and perhaps earlier. Whether Denisovan-related people also reached some of these islands remains uncertain. There are no confirmed Denisovan skeletons from island Southeast Asia. No boat has been found with their name on it. No site has yet produced the kind of unmistakable Denisovan DNA recovered at Denisova Cave. But the genetic inheritance in present-day island populations makes the possibility difficult to dismiss. In the Philippines, some indigenous groups carry especially high levels of Denisovan-related ancestry. This does not prove that Denisovans lived on every Philippine island. DNA can travel through later population movements, marriage networks, and mixing between groups. But it does suggest that the Denisovan story in Southeast Asia was not a brief single encounter. Something more extensive happened. Perhaps modern humans met Denisovan-related groups on the mainland before moving into the islands. Perhaps they met them more than once. Perhaps several Denisovan populations were already scattered across Eastern Asia, each with its own history, and each contributing a slightly different genetic trace to later people. This is where the fossil record and the genetic record seem to be telling stories of very different sizes. The fossil record is small, a cave in Siberia, a jaw on the Tibetan Plateau, a few scattered pieces of bone, tooth, and sedimentary DNA. The genetic record is continental. Denisovan-related ancestry appears, in varying amounts and forms, among populations across East Asia, Southeast Asia, South Asia, Tibet, and Oceania. Some of these signals are faint. Some are clearer. Scientists still debate exactly how many episodes of interbreeding occurred, where they happened, and whether all the DNA came from the same Denisovan population. But the overall pattern is hard to escape. These people, or people closely related to them, occupied a far larger world than their fossils currently reveal. And there are good reasons why their bones may be missing. Siberia is cold, caves can be dry. Under the right conditions, Bone, teeth, and even fragments of ancient DNA can survive for extraordinary lengths of time. Tropical Southeast Asia is almost the opposite. Heat speeds up chemical decay. Humidity encourages microbes. Acidic soils dissolve bone. Dense roots break through old deposits. Heavy rains move sediments downhill, washing away the layers that archaeologists need most. A body buried in a cold cave might leave a bone for hundreds of thousands of years. A body buried in a tropical forest may disappear almost completely. The absence of fossils is not always evidence that people were absent. Sometimes it is evidence that preservation was terrible. Then there is the problem of excavation. Asia is immense. Much of it has never been surveyed systematically for ancient human sites. Caves may be hidden in forested limestone hills, river terraces may lie beneath modern farms and cities. Coastlines used by Ice Age people are now underwater, drowned when the glaciers melted and the seas rose. If Denisovan-related populations lived on the ancient lowlands of Sunda, many of their campsites may now rest beneath the Java Sea, the South China Sea, or other drowned landscapes. And even when archaeologists find human fossils in Asia, identifying them can be difficult. Many bones are fragmentary. Some are old discoveries with uncertain dates or poor excavation records. Others have features that do not fit neatly into modern human, Neanderthal, or familiar earlier human categories. For decades, fossils like these were placed in broad, uncomfortable labels: archaic human, unclassified Homo, possibly related to this group, perhaps related to that one. Now Denisovans have changed the meaning of uncertainty. A strange jaw from China is no longer just a strange jaw. An unusual tooth from a cave in Laos, a fragment from a Chinese hillside, or a skull from an Indonesian island might one day become part of this larger family story. Not every mysterious Asian fossil will turn out to be Denisovan. That would be far too simple. But some may belong to populations we have not yet recognized. And that is the deeper mystery. The Denisovans may not have been a single hidden people waiting to be discovered. They may have been one visible branch of a much broader, more tangled network of ancient Asian human populations. The cave in Siberia gave them a name. Tibet gave them a landscape. The DNA of living people gave them a range stretching toward the warm forests and islands of the southeast. But the spaces between those points are still full of silence, and inside that silence may be not one Denisovan history but several. But nowhere is the Denisovan mystery more striking than on the Tibetan Plateau. This is one of the highest inhabited regions on Earth. Much of it rises more than 12,000 feet above sea level. Some areas are far higher. The air is cold, dry, and thin. Every breath contains the same percentage of oxygen as it does at sea level, about 21%. But the lower air pressure means that each breath delivers fewer oxygen molecules to the lungs, fewer still to the blood, and fewer again to the tissues that need them. For someone arriving from low altitude, this can be miserable. Headaches, nausea, sleeplessness, exhaustion after even gentle movement. In severe cases, fluid can build up in the lungs or brain. The body tries to compensate by making more red blood cells, increasing the amount of hemoglobin available to carry oxygen. That sounds helpful, and at first it is, but too much hemoglobin makes blood thicker. Thick blood is harder for the heart to pump. Over years and generations, a response that saves a visitor can become dangerous for a permanent high-altitude population. This is the problem Tibetan communities solved. Not perfectly, not through one magical gene, not through some simple evolutionary trick, but through a long history of biological and cultural adaptation to a landscape where oxygen is always scarce. And at the centre of that story is EPAS-1. EPAS-1 is part of the body's oxygen sensing system. It helps regulate how the body responds when oxygen levels fall. In many lowland people, chronic exposure to high altitude triggers a powerful increase in red blood cell production. Tibetan populations, on average, often show a different pattern. They can maintain oxygen delivery without pushing hemoglobin levels as high as might be expected. The EPAS-1 variant common among Tibetans appears to play an important role in that response. It does not give people unlimited endurance. It does not make thin air feel like sea level. It does not explain every difference in health, pregnancy, lung function, circulation, metabolism, or development at altitude. But it appears to reduce one of the risks of life in the high mountains, the dangerous overproduction of red blood cells. And when researchers looked closely at this Tibetan version of EPAS-1, they found something extraordinary. Its genetic sequence was very unusual compared with most versions found in living humans. But it was strikingly similar to the sequence recovered from Denisovan DNA. Not vaguely similar. Not merely a distant family resemblance shared by all ancient humans. Similar enough that the strongest explanation is inheritance through interbreeding. At some point, ancestors of present-day Tibetans met and had children with Denisovan-related people. Those children inherited pieces of Denisovan DNA. Most of those inherited pieces may have done nothing especially useful. Over thousands of generations, many would have become rarer, broken apart by recombination, or disappeared entirely. But EPAS-1 was different. On the plateau, where low oxygen shaped every pregnancy, every childhood, every winter journey, every hunt, and every season of herding, this Denisovan-related variant seems to have offered an advantage. Natural selection favoured it. Generation after generation, it became more common. This is one of the clearest known examples of archaic human ancestry contributing directly to a modern human adaptation. A gene inherited from a vanished population is still helping people live in one of the most demanding environments on Earth. But even that sentence needs care. The Denisovans did not simply hand Tibetans a finished survival package. High-altitude adaptation involves many genes, not one. Other genetic variants, including changes associated with oxygen regulation and blood flow, also matter. So do the effects of development. A person raised from childhood at high altitude may develop differently from someone who arrives as an adult. And culture matters too. Warm clothing, shelter, food storage, trade routes, animal herding, knowledge of weather, and social support can all make survival possible where biology alone might not be enough. Human adaptation is rarely just genetic. It is bodies, behaviour, knowledge, community and landscape, all working together. Still, EPAS-1 tells us something important. Denisovan-related people were not merely passing through Asia as occasional strangers encountered by migrating modern humans. At least some populations had lived long enough in or near high-altitude environments for natural selection to shape their biology. And the strongest archaeological clue comes from Baishia Karst Cave. This cave lies on the northeastern edge of the Tibetan Plateau, more than 10,000 feet above sea level. It is not a place where a lost traveller would casually wander. The winters are severe. The growing season is short. The terrain is steep and exposed. Even today, living there requires planning and experience. In 1980, a Buddhist monk found a large fossil jaw in the cave. For decades it sat in collections without a clear place in the human family tree. It had no DNA available for analysis. But researchers later recovered ancient proteins from one of its teeth. Those proteins showed that the Jahe jaw belonged to a Denisovan-related individual. The jaw was ancient, probably at least 160,000 years old. That alone changed the map. Then sediment samples from the cave added another layer. Researchers found Denisovan DNA preserved not in a skeleton, but in the soil itself. tiny genetic traces left behind by bodies that had decayed long ago. The evidence suggests Denisovan-related people visited, occupied, or repeatedly used the cave over a very long span of time. The exact pattern is still being worked out. Sedimentary DNA is powerful, but it cannot tell us everything. A trace in the soil does not reveal whether the people stayed for days, months, seasons, or generations. It does not show us their faces, their language, or the shape of their social world. But the pattern is difficult to ignore. Denisovan-related people were on the Tibetan Plateau long before modern humans are known to have become established there. They were not just surviving a brief high-altitude emergency. They may have been returning to this landscape again and again, across changing climates and long stretches of deep time. Imagine what that required. They would have needed to find food in a sparse environment. Wild yak deer, smaller mammals, perhaps birds, and plant foods when seasons allowed. They would have needed fire, shelter, clothing, and intimate knowledge of a landscape that could turn lethal without warning. They would have learned which valleys offered protection, which routes stayed open after snow, where animals moved, and when storms made travel impossible. No written record survives, no Denisovan campfire story has reached us. But their descendants may have left one small message in the blood of living people, a message that says they knew the thin air, and that perhaps they helped later humans learn to live there too. But the Tibetan Plateau raises another question. If Denisovan-related people could live in Siberia, on the high plateau of Tibet, and perhaps across other parts of Asia, were they really one population at all? For a long time it was easy to imagine them that way. There was Denisova Cave in the Altai Mountains. There was a finger bone, a few teeth, a jaw from Tibet, a handful of genetic clues scattered through living people. The simplest picture was a small, isolated human group, living somewhere in Central or Northern Asia, occasionally meeting Neanderthals and modern humans at the edges of its territory. But DNA has made that simple picture increasingly difficult to keep. The Denisovan genome recovered from Denisova Cave is one of the most important ancient genomes ever sequenced. It gave researchers a direct genetic glimpse of an individual who lived in southern Siberia tens of thousands of years ago. This Altai Denisovan became the reference point. When scientists found Denisovan-like DNA in living people, they compared it to her genome. And at first, the results seemed straightforward. Some modern populations, especially in East and Southeast Asia, carry small amounts of DNA that resemble the Altai Denisovan genome more closely than ordinary modern human DNA does. Tibetans, as we have seen, carry a Denisovan-related version of EPAS1. Other populations across Asia carry additional traces, small fragments passed down through countless generations. But then researchers looked at the peoples of Oceania, and the story became much stranger. People in Papua New Guinea, Aboriginal Australians, and some other populations in island Southeast Asia and the Pacific can carry substantially more Denisovan ancestry than most mainland Asian populations. In some Papuan populations, the total may reach several % of the genome. That is already remarkable. A vanished human group, known from a cave in Siberia and a few fossil fragments elsewhere, has left a larger genetic legacy in people living thousands of miles away, on islands near the edge of the Pacific. But the deeper surprise was this. Much of the Denisovan ancestry in Oceania does not look like it came directly from the same population represented by the Altai Denisovan genome. It is related, clearly related, but not identical. Imagine finding an old family photograph in a house in Siberia, then discovering distant cousins in New Guinea, whose inherited features suggest they came from another branch of the same family, one that separated from the Siberian branch long before either branch met modern humans. That is roughly what the genetics suggests. The Altai Denisovan was not necessarily the ancestor of the Denisovan-related people, who contributed DNA to Papuan ancestors. She may have belonged to a northern population, perhaps one adapted to the forests, mountains, and cold seasonal world of southern Siberia. The group that met the ancestors of Papuans may have lived much farther south, perhaps in tropical mainland Asia, island Southeast Asia, or regions now hidden beneath forests, coastlines, and rising seas. And these populations may have been separated for a very long time. Not years, not centuries, potentially hundreds of thousands of years. That does not mean they were different species in the neat modern sense. Ancient human populations did not always divide into clean, permanent categories. They separated, met again, exchanged genes, moved into new landscapes, and separated once more. The boundaries were probably messy. The family tree often looked less like a tree than a braided river, with channels splitting apart and occasionally flowing back together. Still, the genetic differences matter. They suggest that Denisovan diversity may have been substantial. There may have been northern Denisovans, southern Denisovans, high-altitude Denisovans, and perhaps other populations we have not yet detected. Some may have been closely connected. Others may have lived so far apart for so long that their genomes became distinctly different. Asia is enormous. It contains Arctic cold, temperate forests, deserts, river valleys, tropical rainforests, mountain plateaus, coastlines, and island chains. A human population spreading across that continent would not remain unchanged. A group living through Siberian winters faced one set of pressures. A group living in the Himalayan region faced another. A group surviving in humid tropical forests, where plants, parasites, animals, and seasonal rains shaped daily life, would face another entirely. Over thousands of generations, natural selection, isolation, and chance could produce very different Denisovan populations. And if you enjoy these moments where one discovery opens into 10 deeper mysteries, the channel's book about 12 unexplained mysteries explores more of them. You can find it through the website link in the description. The evidence for multiple Denisovan populations does not come from one single genetic result. It comes from patterns. Different living populations carry Denisovan fragments that do not all match one another equally well. Some fragments are more similar to the Altai Denisovan. Others appear to come from a more deeply separated Denisovan-related lineage. Studies have proposed more than one episode of interbreeding between modern humans and Denisovan populations, occurring at different times and in different places. The exact number is still debated. Scientists do not all agree on how many Denisovan populations existed, where they lived, or how often they met modern humans. Genetic models depend on assumptions. Ancient DNA is incomplete. Modern DNA has been shuffled by migration and recombination for tens of thousands of years. Every generation breaks inherited fragments into smaller pieces, making the original encounters harder to reconstruct. But the broad conclusion is becoming harder to avoid. There was probably no single Denisovan homeland. Denisova Cave was not necessarily the center of their world. It may simply be one rare place where cold, dry conditions preserved enough DNA for us to find them. Think about how much must be missing. A cave in Siberia preserves a genome. A cave on the Tibetan Plateau preserves a jaw and traces in the soil. But vast areas of Asia have yielded almost nothing. Tropical climates destroy DNA quickly. Dense forests hide sites. Rising seas have drowned ancient coastlines where people may once have lived. Rivers erode campsites. Time grinds bone into dust. The Denisovans we know may be only the survivors of preservation. Not the survivors themselves, the survivors of evidence. Somewhere in southern Asia, perhaps, there may once have been a population whose DNA now survives mainly in the people of New Guinea and the Pacific. Somewhere in East Asia, Another Denisovan-related group may have met the ancestors of later populations and left smaller genetic traces behind. On the Tibetan Plateau, a population adapted to thin air may have passed on one of the most useful inherited variants known from any archaic human group. One name, Denisovan, may be hiding many histories, and that changes the emotional shape of the story. They were not necessarily a tiny band of mysterious cave dwellers at the edge of Siberia. They may have been a widespread network of populations scattered across much of Asia, each adapting to its own landscape, each carrying its own version of a shared ancient inheritance. Then modern humans arrived in greater numbers, and in some places they did not simply replace the Denisovans. They met them. What did that meeting look like? We do not know. There is no preserved scene of it, no Denisovan camp beside a modern human camp, no written account, no name for the river valley, the forest path, or the shoreline where two groups first saw one another. What we have instead is quieter, and in some ways more intimate. We have DNA. Every person carries a genome assembled from countless ancestors. Most of those ancestors are impossible to identify. Their bones are gone, their languages are gone, their homes have collapsed into soil. But occasionally, a piece of DNA survives the passage of time, moving from parent to child, generation after generation, until it reaches people living today. And in the genomes of many modern people, scientists have found pieces that did not come from the main line of Homo sapiens ancestry. They came from Denisovan-related populations. This is not a vague resemblance. Researchers can compare sections of modern DNA with the high-quality Denisovan genome from Denisova Cave, along with Neanderthal genomes and DNA from present-day African populations. When a segment in a living person is unusually similar to Denisovan DNA and cannot easily be explained by shared ancestry alone, it points toward an ancient encounter. A meeting, a relationship, children born into families whose ancestors had lived apart for hundreds of thousands of years. The clearest signal appears in Oceania. People from Papua New Guinea, along with some Aboriginal Australian populations and other populations in parts of island Southeast Asia and the Pacific, carry comparatively high levels of Denisovan ancestry. In Papuan populations, Denisovan-derived DNA can account for several percent of the genome, though the exact amount varies between individuals and communities and depends on the methods used to measure it. Several percent may not sound like much, but think about what it means. After tens of thousands of years, after every generation mixed, moved, divided, and recombined its DNA, a detectable part of the Denisovan inheritance remains. Not in one isolated family, not in a single village, but across populations whose ancestors travelled through some of the most difficult landscapes on Earth: tropical forests, mountain chains, river systems, island passages, coasts that have since been swallowed by rising seas. The ancestors of Papuan peoples did not receive this ancestry from a single Siberian woman in Denisova Cave. That much is almost certain. The genetic patterns suggest they encountered a southern Denisovan-related population, one genetically distinct from the Altai Denisovan whose finger bone first revealed this lost branch of humanity. Somewhere along the route into Southeast Asia and Sahul, the ancient landmass that once joined New Guinea and Australia, modern humans met Denisovan-related people, and perhaps they met them more than once. This is where the story becomes less like a single migration and more like a long journey through occupied territory. For years, the popular picture of modern human expansion was simple. Homo sapiens leaves Africa. Homo sapiens spreads across Asia. Other human groups disappear. But the genome tells a messier story. Modern humans leaving Africa around 50,000 to 70,000 years ago were not moving through an empty world. Neanderthals lived across western Eurasia. Denisovan-related populations appear to have occupied parts of Asia perhaps from high mountain regions to warmer southern lands. Other archaic populations may also have existed, leaving genetic traces too faint or too complicated for us to identify clearly. The first modern humans entering Asia may have encountered different Denisovan groups at different points along their journey. Some contact may have happened on the mainland, perhaps in South Asia, East Asia, or Southeast Asia. Some may have occurred farther east among the islands. Some genetic studies suggest that the ancestors of Oceanian populations received Denisovan ancestry in multiple pulses, from populations that were themselves genetically different from one another. The exact map remains uncertain, but the implications are striking. If modern humans acquired Denisovan ancestry before reaching New Guinea and Australia, then Denisovan-related populations may have lived somewhere along the southern dispersal route. If other populations in East Asia carry different Denisovan signals, then encounters may also have occurred farther north or farther inland. The routes taken by early humans were not one straight arrow leaving Africa. They were branching paths, pauses, returns, coastal journeys, mountain crossings, and generations spent adapting to unfamiliar worlds. And the Denisovans were part of that world. Smaller Denisovan-derived signals appear in populations across Asia. They have been detected at varying levels and in varying forms among populations in East Asia, Southeast Asia, and the Himalayan region. People in the Americas also carry small amounts indirectly, largely because the ancestors of indigenous American populations came from Northeast Asian populations that already had some Denisovan-related ancestry. These patterns do not mean that every population had a direct meeting with Denisovans. Human groups moved repeatedly. They mixed with neighbouring groups. Genes travelled long after the original people who carried them had vanished. A Denisovan fragment found in one modern population may reflect an encounter far away, thousands of years earlier, inherited through many later migrations. DNA records ancestry. It does not preserve a passport stamp. And this matters because genetic percentages can be misunderstood. To say that a population has Denisovan ancestry is not to say that its people are partly Denisovan in some fixed cultural or personal sense. It does not define identity. It does not determine language, community, history, or belonging. These fragments are records of deep population history, inherited unevenly and reshaped by recombination over immense spans of time. Two people from the same community may carry slightly different archaic fragments. One may inherit a Denisovan segment that another does not. Over generations, some fragments disappear by chance. Others become more common. Natural selection may preserve a useful variant, while most of the surrounding DNA fades away. So these percentages are not labels, they are echoes. And they reveal something important about those first encounters. Modern humans did not always respond to other human groups with immediate violence, avoidance, or replacement. There may have been conflict. Competition for food, territory, and shelter was real. Small populations living in harsh environments could not always afford peaceful coexistence. But interbreeding happened, repeatedly enough to leave a visible mark in living genomes. That means, at least sometimes, people crossed the boundaries between groups. They formed relationships. They raised children. Those children survived. Their descendants joined later communities, carrying a few Denisovan fragments onward into the future. The great human expansion across Asia was not simply a story of one people replacing another. It was also a story of meeting, mixing, and carrying something forward. And in one cold cave in southern Siberia, science found the most direct proof of just how close those meetings could become. And then the cave gave us something even more extraordinary. Not a distant genetic echo preserved in living people. Not a faint statistical pattern spread across thousands of genomes. A child, in 2012, Researchers examining material from Denisova Cave identified a tiny fragment of long bone. It was small enough to fit in the palm of a hand. At first, there was almost nothing remarkable about it. The fragment could not tell archaeologists what the person's face looked like. It could not reveal her height, her voice, or the language spoken around her. But it still contained DNA, and when scientists sequenced that DNA, they found one of the most intimate discoveries in all of human prehistory. The bone belonged to a girl, probably at least 13 years old when she died. Researchers gave her the laboratory name Denisova 11. Sometimes she is also called Deni. Her mother was Neanderthal. Her father was Denisovan, not mostly Neanderthal, with some Denisovan ancestry from a distant ancestor. Not mostly Denisovan, carrying a faint Neanderthal fragment inherited centuries earlier. This was the direct genetic signature of a first-generation child. Half Neanderthal, half Denisovan. A family reunion in a cave. Think about how astonishing that is. For years, scientists knew that Neanderthals, Denisovans, and modern humans had interbred. The evidence was written in living genomes. People outside Africa usually carry some Neanderthal ancestry. Many populations in Asia and Oceania carry Denisovan-derived ancestry. The broad conclusion was already clear. Different human groups had met and sometimes had children together. But Denisova 11 made the abstract personal. Her parents were not populations on a graph. They were two individuals at some point, a Neanderthal woman and a Denisovan man, met closely enough to form a family. Their daughter lived long enough for part of one of her bones to end up buried in the sediments of Denisova Cave, where it survived for tens of thousands of years until modern science found it. There is something almost unsettling about the scale of that survival. An adolescent girl lives in Ice Age Asia. Her parents come from populations that had been evolving along separate paths for hundreds of thousands of years. She dies. Her body disappears into the ground. Nearly 100,000 years pass. Empires rise and collapse. Languages appear and vanish. And then a few cells inside one small bone fragment reveal that her parents belonged to groups we had once imagined as separate branches, isolated from one another in the deep past. They were not completely isolated. They could meet. They could mix. They could have children, and those children were genetically viable. The details become even more interesting when scientists looked beyond the simple half-and-half result. Denisova 11's mother was Neanderthal, but she was genetically closer to Neanderthals known from Europe than to an earlier Neanderthal population represented in the same Siberian cave. This suggests that Neanderthal populations moved across Eurasia, replacing or mixing with earlier groups over time. Her Denisovan father also carried a small amount of Neanderthal ancestry, inherited from an even earlier encounter somewhere in his own family history. So Denisova 11 was not standing at the first crossing between two worlds. She was part of a longer pattern. Her father's ancestors had already received Neanderthal DNA. Her mother belonged to a Neanderthal population with connections stretching far to the west. And she herself carried an almost perfectly balanced inheritance from both groups. The ancient human family tree was not a clean tree at all. It was a braided river, branches split apart. They travel in different directions. They adapt to different climates and landscapes. Then, sometimes, the channels meet again. Water passes between them. The boundaries blur, and farther downstream it becomes difficult to say where one current ended and another began. Still, one bone cannot answer every question. Denisova 11 proves that a Neanderthal and a Denisovan had a child. It does not prove that such relationships were common everywhere, or that all Neanderthal and Denisovan groups mixed regularly. One extraordinary individual cannot tell us how often families like hers formed, whether they were accepted by their communities, or what circumstances brought her parents together. Maybe these encounters were rare, happening only where small populations overlapped along shifting frontiers. Maybe they were more frequent than we once thought, especially in places like the Altai Mountains, where different groups could have been drawn to the same sheltered valleys, hunting grounds, stone sources, and cave systems. The fact that this first-generation hybrid appeared in such a small sample is certainly striking. Scientists had recovered DNA from only a limited number of ancient individuals when Denisova 11 was identified. Finding someone with one Neanderthal parent and one Denisovan parent so quickly suggests that contact between these groups may not have been impossibly rare. But striking is not the same as certain. The Denisova Cave record is incomplete. The people whose bones survived may not represent the full population. The cave itself may have been an unusual place, used repeatedly by different groups precisely because it sat within a landscape where routes crossed. What we can say is simpler and more powerful. The boundaries between ancient human groups were real, but they were not walls. Neanderthals and Denisovans had different evolutionary histories. They likely looked somewhat different. They carried different genetic adaptations. They may have had different traditions, different territories, perhaps even different ways of speaking and organizing their lives. Yet none of that prevented every encounter. At least once, and probably more than once, a Neanderthal woman and a Denisovan man became the parents of a child. That child was Denisova 11, and her existence changes the feeling of the whole story. The Denisovans were never just a ghost population, reconstructed from a finger bone and a few percentages in modern DNA. The Neanderthals were never simply another extinct species locked away in Europe. In the cold interior of Asia, their lives overlapped. Their families overlapped. Their histories became entangled in ways that no stone tool or skull shape could fully explain. And all of it happened in one place that seems to have drawn human groups together again and again. Denisova Cave was not merely where scientists found their remains. It may have been a meeting ground, but what does it really mean to call Denisova Cave a meeting ground? It does not mean Neanderthals, Denisovans, and modern humans were all standing around the same fire on the same winter evening. The evidence does not allow that kind of certainty. The cave was used over an immense stretch of time. People came and went. Sediments accumulated slowly beneath their feet. Roof fragments fell. Animals carried bones inside. Water moved fine particles through the chambers. A layer that looks like a single band of earth can represent thousands of years. Still, when scientists put together the bones, DNA, artifacts, and dates, a remarkable pattern appears. Denisovans used the cave, Neanderthals used the cave, and later modern humans used the cave too, not necessarily as neighbours, not necessarily as enemies, but all three groups passed through the same mountain landscape and returned at different moments to the same sheltered place. Denisova Cave lies in the Altai Mountains of southern Siberia, near the place where modern Russia, Mongolia, Kazakhstan and China come close together. It is not an easy environment. Winters can be severe. The landscape shifts from forest to open steppe, from river valleys to high ridges. But that difficulty may be exactly what made the region important. It was a crossroads. From the west came routes leading toward Central Asia and Europe. From the east came paths into Mongolia and northern China. To the south lay the approaches to the Tibetan Plateau and the vast interior of Asia. Animals moved through these corridors. So did people, and a cave in a well-positioned valley could offer shelter, a stable place to return to, and access to hunting grounds, water, firewood, and sources of stone. For ancient humans, it may have been less like an isolated home at the end of the world, and more like a familiar stop along a difficult road. The chronology of the cave has become much clearer in recent years, though not perfectly clear. That improvement came from a scientific problem that sounds simple but is actually incredibly difficult. How do you date dirt? Bones can sometimes be dated directly, especially if they still contain enough collagen for radiocarbon dating. But Denisova Cave is older than the reliable range of most radiocarbon methods in many of its deepest layers, so researchers have had to use several techniques together. They have dated tiny mineral grains in the sediments using a method called optically stimulated luminescence. Basically, these grains record how long it has been since they were last exposed to sunlight. Scientists have also used radiocarbon dating where possible, uranium series methods on cave deposits, and genetic evidence from the fossils themselves. The result is not one clean timeline. It is a carefully reconstructed sequence with ranges of dates rather than a single perfect answer. Denisovans appear to have occupied the cave at multiple points, perhaps beginning more than 200,000 years ago and continuing until roughly 50,000 years ago, though the exact timing of every visit remains debated. Neanderthals also appeared there, including the population connected to Denisova 11's mother. Their presence seems especially associated with colder periods, when conditions in the Altai may have favoured groups adapted to the harsher climates of northern Eurasia. Then, much later, around 45,000 years ago, or somewhat earlier, modern humans entered the picture. This is where the cave becomes even more mysterious. The upper layers of Denisova Cave contain objects that seem unusually sophisticated for their age. There are carefully made stone tools. There are bone tools. There are ornaments, including a polished greenstone bracelet and beads made from animal teeth, bone, and other materials. Some of these objects are among the earliest known examples of such personal ornaments in northern Eurasia. For a long time it was tempting to connect these beautiful artifacts directly to the Denisovans. After all, this was Denisova Cave. The Denisovans were the human group that made the site famous. Maybe they made the bracelet? Maybe they drilled the beads. Maybe they were creating symbolic objects and complex technologies that rivaled anything made by early modern humans. It is possible, but possible is doing a lot of work there. An artifact found in a cave layer is not automatically made by the human bone found nearby. A cave can be used by different populations across thousands of years. Objects can shift slightly through disturbed sediment. A later visitor can occupy the same chamber as an earlier visitor. A stone tool can remain on the ground long after the people who made it are gone. And at Denisova Cave, the human fossils are frustratingly sparse. A finger bone, teeth, fragments of bone, the remains of Denisova 11. Enough to transform human evolution, but not enough to attach a name to every tool, ornament, or hearth. This is one of archaeology's most persistent problems. We want to ask, who made this? Neanderthal, Denisovan, or modern human? But the honest answer is often, we do not know. Some of the upper layer ornaments and tools are now generally linked to early modern humans because their dates overlap with the arrival of Homo sapiens in the region. Yet even there, the story may not be simple. Modern humans could have made the objects themselves. They could have learned techniques through contact with other groups. Or the layers may preserve a more complicated sequence of changing occupations than any single label can capture. The cave gives us evidence of proximity. It does not always give us ownership. What it does show very clearly is that the Altai was not a permanent homeland belonging to just one humankind. It was a zone of shifting occupation. During warmer phases, forests and mixed landscapes expanded. Animal communities changed. Routes through valleys may have become easier. During colder, drier periods, open steppes spread across the region, and groups with different hunting strategies and cold weather adaptations may have moved in or moved away. Climate did not simply make life hard, It rearranged the map. A warming period could open a corridor, bringing distant populations into contact. A cold phase could isolate groups in separate refuges. Changes in rainfall could alter river systems. Changes in vegetation could shift the migrations of deer, bison, horses, and other prey. Human groups followed resources, and in following them they sometimes followed one another. That may be why Denisova Cave holds such an extraordinary mixture of evidence. Not because it was magical, not because every ancient human group gathered there intentionally, but because it stood in a landscape repeatedly reshaped by climate, migration, and survival. A place where different branches of humanity could cross paths, and if the cave was a meeting ground, then its tools and ornaments are not just objects. They are clues from a crowded, complicated world where we still cannot always tell whose hands held them. So let's stay with those objects for a moment, because they are some of the most beautiful and frustrating clues in the entire Denisovan story. In the upper layers of Denisova Cave, archaeologists found an assemblage that looks very different from the simpler stone technologies of much earlier periods. There are long, carefully shaped blades. There are burins, tools with sharp chisel-like edges used for working bone, antler, wood, or hide. There are scrapers, points, and small retouched implements designed for particular jobs rather than just smashed into usefulness. This is what archaeologists call an Upper Paleolithic toolkit. And it suggests planning. To make a long blade from stone, you do not just hit a rock and hope for the best. You choose the right raw material. You prepare the core. You strike it at the right angle, with the right amount of force. You need to understand that the stone contains possible tools before those tools even exist. That is abstract planning, it is manufacturing, in the ancient sense of the word. Then there are the bone tools. Some were shaped into points, possibly used as awls, needles, or parts of hunting weapons. Bone is harder to work than stone in some ways, but it can be shaped into thin, durable forms that stone cannot easily produce. A bone point can pierce hide, it can help sew clothing. And in Ice Age Siberia, where winter temperatures could fall far below freezing, good clothing was not a luxury. It was survival. The people who made these tools understood their materials. They knew stone, they knew bone, they knew which animal parts could become useful objects after a hunt was over. But the ornaments are where Denisova Cave becomes truly remarkable. There are beads made from animal teeth. There are pendants made from bone and ivory. There are pierced objects that may once have hung from clothing, hair, belts, or necklaces. These were not necessary for cutting meat or building shelter. They had another purpose. They made a statement. Maybe they marked identity. Maybe they showed membership in a family, a group, or a trading network. Maybe they carried memories of a successful hunt, a distant journey, or someone who had died. We cannot know the exact meaning, but ornaments tell us that ancient people were doing more than meeting basic needs. They were shaping how they appeared to other people. They were making objects that carried social meaning. And then there is the bracelet. It is made from a dark green stone, probably chloratolite, brought to the cave from some distance away. The stone was carefully shaped, drilled and polished until its surface became smooth and almost luminous. It is not a rough pebble with a hole punched through it. It is a deliberately crafted object, made with skill and patience. Imagine the work involved, finding the right stone, breaking it into a usable piece without ruining it, grinding and shaping it, drilling a hole with a rotating tool and abrasive grit, polishing the surface again and again. This was not quick work. The bracelet has often been presented as evidence that Denisovans were makers of sophisticated ornaments, perhaps even artists. and that idea is understandable. The bracelet came from Denisova Cave. Denisovans gave the cave its name. It is tempting to put the object in Denisovan hands. But archaeology has a rule that can be deeply unsatisfying. Found together does not always mean made together. The bracelet and many of the other sophisticated artifacts came from layers generally dated to around 45,000 years ago, give or take. That is roughly the period when modern humans were expanding across northern Eurasia. It is also later than the best established Denisovan remains from the cave. So, who made them? One possibility is that early modern humans made the blade tools, bone points, beads, and bracelet after arriving in the Altai. This is currently the most widely accepted interpretation for much of the Upper Paleolithic material. Similar technologies and ornaments appear at other sites connected to early Homo sapiens across Eurasia. But that does not close the case. Denisovans may have survived in the region later than the fossils currently show. Their DNA appears in cave sediments from multiple periods, and sediment DNA has expanded the record beyond the tiny number of bones that can be identified by eye. A people do not need to leave a skull or a skeleton behind every time they enter a cave. Sometimes they leave only a few cells, fragments of DNA trapped in dirt that no ancient visitor could have imagined becoming evidence. It is possible that Denisovans made some complex objects of their own. It is possible that Neanderthals, who also moved through this region, contributed particular traditions. It is possible that modern humans arrived with their own skills, but adopted ideas, materials, or techniques through contact with the groups already living in Asia. And it is possible that a tool tradition does not belong neatly to one species at all. That is important. We often imagine culture as a label. Neanderthal tools, Denisovan ornaments, modern human art. But people learn from one another. They copy useful techniques. They trade materials, they marry across group boundaries. And in the Altai we know this was not just speculation. Denisova 11 was the child of a Neanderthal mother and a Denisovan father. Her existence proves that boundaries between these populations were real, but not absolute. If people could have children together, they could have exchanged knowledge too. A skilled toolmaker might show someone how to prepare a stone core. A hunter might trade a rare piece of green stone. An ornament style might spread from one group to another because it was admired, useful, or socially meaningful. By the time archaeologists find the finished object, thousands of years later, the cultural trail may be impossible to reconstruct. Genetics can tell us who was present. Archaeology can tell us what was made. But joining those two answers together is much harder. At Denisova Cave, the artifacts reveal a world of technical skill, long-distance movement, and symbolic behavior. The DNA reveals a world of multiple human populations meeting, mixing, and disappearing. Together they give us a richer picture than either could provide alone. But they also leave us with an uncomfortable truth. The most famous bracelet in Denisovan history may not have been made by a Denisovan. Or it may have been, and that uncertainty is not a failure of science. It is the honest shape of the evidence, and that uncertainty follows us out of the cave. Because the most important Denisovan evidence is not stored in a museum drawer. It is not buried beneath Siberian soil. It is living. It is carried in the DNA of people alive today. When modern humans moved through Asia, they did not simply replace every population they encountered. In some places they met Denisovans. They had children together. Those children had descendants, and some of those descendants passed small pieces of Denisovan DNA forward, generation after generation, through thousands of years of migration, climate change, famine, disease, and survival. Most people outside Africa carry some Neanderthal ancestry. Denisovan ancestry has a different pattern. It is especially noticeable in parts of Asia and Oceania. People in New Guinea, Australia, and some island Southeast Asian populations can carry several percent Denisovan-related ancestry, although the exact amount varies between populations and depends on how scientists measure it. In mainland Asia, the Denisovan contribution is often smaller. But small does not mean unimportant. A single inherited genetic segment can sometimes affect how the body responds to a particular environment. And the most famous example comes from one of the hardest places humans have ever made a home, the Tibetan Plateau. At altitudes above 13,000 feet, the air contains far less oxygen than it does at sea level. For someone not adapted to it, living there can be punishing. The body struggles to get enough oxygen into the blood. Pregnancy becomes more difficult. Physical work becomes exhausting. In severe cases, the response to thin air can become dangerous, as the body produces too many red blood cells and makes the blood thicker. Yet, Tibetan populations have lived on the plateau for thousands of years. One reason appears to involve a gene called EPAS1. EPAS1 helps regulate the body's response to low oxygen. In Tibetans, one version of this genetic region is strongly associated with a more efficient high-altitude response. Rather than simply flooding the blood with extra red cells, the body can manage oxygen stress in a different way. And when scientists compared this unusual genetic segment with ancient genomes, they found that it looked far more like Denisovan DNA than the DNA of other modern human populations. This is one of the clearest cases we have of Denisovan-derived DNA being favoured by natural selection in living humans. Think about what that means. At some point in the deep past, modern human ancestors living near or on the highlands encountered a Denisovan population already adapted, or partly adapted, to thin air. Through interbreeding, a useful version of this gene entered the modern human population. Then, over many generations, people carrying that version may have had a better chance of surviving and raising children in the oxygen-poor world of the plateau. The Denisovans themselves are gone, but a small piece of their biological history may still help millions of people breathe at high altitude. That is extraordinary, but it is also exactly the kind of thing evolution does. It does not care where a useful genetic variant came from. If a gene helps its carrier survive in a particular environment, it can spread. A population can inherit a solution rather than inventing one from scratch. Scientists look for this process by searching archaic DNA segments for signs of natural selection. They ask questions like these. Is a Denisovan-like segment unusually common in one modern population? Is it much rarer elsewhere? Is the segment longer than expected, suggesting that it entered the population relatively recently and spread quickly? Does it occur near genes involved in oxygen use, immunity, metabolism, or skin and hair biology? If the answers line up, researchers may have evidence that natural selection preserved that ancient inheritance. But may is an important word here. Not every piece of Denisovan DNA was useful. Much of it may have been neutral, doing no particular harm and no particular good. Some inherited segments may once have helped but no longer matter in a changed environment. Others may have caused problems and gradually disappeared from the human gene pool. In fact, much of the story of archaic ancestry is loss. After interbreeding, modern human genomes contained many Neanderthal and Denisovan segments. Over time, recombination broke those segments into smaller pieces. Natural selection removed some variants that were poorly matched to modern human genetic backgrounds. Chance erased others. What remains is not a complete Denisovan genome scattered among living people. It is a patchwork, a few surviving paragraphs from a book whose rest has burned away. Some of those paragraphs may concern immunity. This makes sense. When populations move into new regions, they encounter unfamiliar pathogens. Viruses, bacteria, parasites, all the invisible dangers that come with new landscapes and new animal populations. Genes involved in immune responses are often shaped by strong selection, because disease can kill quickly and because immunity is a constant evolutionary arms race. Researchers have identified Denisovan-like genetic contributions near immune-related regions in some modern populations. There are also possible Denisovan-derived variants connected with metabolism, fat storage, and the way bodies respond to cold or changing diets. In Arctic populations, for example, a Denisovan-like region around genes called TBX15 and WARS2 has been studied for possible links to body fat distribution and metabolic adaptation. But this is where the story becomes less tidy. A gene associated with a trait is not automatically a gene that causes that trait. Modern traits are usually complicated. Height, diabetes risk, immune activity, fat distribution, athletic performance, fertility, even the way a body reacts to cold, all involve many genes interacting with food, disease, exercise, stress, culture, and plain old chance. Finding an ancient DNA segment near a gene is a clue. Finding that the segment is common in a population is another clue. But neither clue by itself proves that Denisovan ancestry created a modern trait. Sometimes the inherited segment may simply be travelling alongside the real causal variant. Sometimes a genetic effect found in one study becomes weaker when tested in another population. Sometimes scientists can identify the Denisovan-like DNA, but cannot yet say what it does at all. And that is not unusual. The human genome is enormous. We have learned to read its letters faster than we have learned to understand its meaning. Still, the broad fact remains. Deep prehistory is not completely dead. It is present in blood chemistry, in oxygen responses, in immune systems, in tiny molecular switches that turn genes up or down at different moments in life. Every modern genome is a record of ancient meetings, ancient migrations, and ancient families. A Tibetan child born today may carry a genetic inheritance shaped by people who lived on the high plateau tens of thousands of years ago. A person in New Guinea may carry more Denisovan ancestry than anyone once thought possible, from a population known only through a finger bone and a few teeth. Somewhere inside living bodies are fragments of encounters that no one wrote down between people whose faces we can barely reconstruct. The Denisovans did not vanish without a trace. They vanished as a distinct people, but parts of them continued onward, carried by the children of those encounters, and by the children of their children all the way into the present. But even that is not the strangest part. The Denisovan DNA carried by living people does not all point back neatly to the Denisovan girl from Siberia. At first, scientists assumed it might. After all, her tiny finger bone had given researchers the first Denisovan genome. It was the reference point, the genetic face of a population we could barely see. So when Denisovan-like DNA appeared in modern people from New Guinea, Australia, the Philippines, Tibet, and across parts of Asia, the obvious question was simple. How closely did those surviving fragments match the Altai Denisovan? The answer was surprising, often not very closely. They were related, clearly related, but some Denisovan ancestry in living populations appears to come from groups that had been separated from the Altai Denisovans for a very long time. Not a few generations, not a few centuries, potentially hundreds of thousands of years. This is where the story becomes less like a family tree and more like a forest in the dark. Geneticists have a name for populations like these. Ghost populations. A ghost population is not imaginary. It is a real population inferred from the traces it left behind, even though we have not found its bones, its teeth, its tools, or its DNA in an ancient fossil. It is a population detected indirectly, the way you might know someone crossed fresh snow, because you see footprints leading into the trees, even if you never see the person who made them. In genetics, the footprints are patterns of shared mutations. Researchers compare the DNA of living people with ancient genomes from Neanderthals, Denisovans, and early modern humans. They look for stretches of DNA that are too different to have come from ordinary variation within modern humans, but also too different to be a close match for the known Altai Denisovan genome. When those strange segments appear again and again, especially in particular populations, they suggest a missing source. Something was there, someone was there, and that someone may have been Denisovan, or at least closely related to Denisovans, without being genetically identical to the people who lived near Denisova Cave in Siberia. This matters because the Altai Mountains are cold, dry, and unusually kind to ancient DNA. A small bone can survive there for tens of thousands of years, protected in a cave where temperatures remain low and stable. Tropical Asia is a completely different world. Heat, humidity, acidic soils, insects, bacteria, and constant rainfall are brutal to bone and devastating to DNA. A skeleton can vanish into the soil, a genome can break apart long before any archaeologist arrives. The very regions where some of the strongest Denisovan signals survive in living people, Ireland, Southeast Asia, New Guinea, and perhaps parts of southern China, are also among the hardest places on Earth to recover ancient DNA. So the fossil record may be giving us a distorted map. It shows us the places where remains happen to survive, not necessarily the places where Denisovans were most numerous, most diverse, or most important. Imagine trying to reconstruct the history of an entire continent from a few scraps of paper that escaped a fire. One page is found in Siberia, another in Tibet. A tooth appears in Laos. Then modern genomes from thousands of people reveal that the missing chapters were probably scattered across forests, islands, river valleys, and mountain ranges from northern Asia to Oceania. The Denisovans may not have been one small isolated population. They may have been many populations spread across enormous distances, adapting to radically different environments. High mountain plateaus, northern forests, tropical lowlands, coastal regions, islands, separated by dangerous stretches of water. Some groups may have been genetically close to the Altai Denisovans. Others may have been distant cousins, separated for so long that calling them all one single population becomes misleading. And modern human ancestors may have met more than one of them. Genetic studies increasingly suggest that there were multiple episodes of Denisovan-related interbreeding. The ancestry found in Papuan and Australian populations does not appear to have exactly the same history as the ancestry found in East Asians. The Denisovan-like DNA associated with Tibetan high-altitude adaptation also has its own peculiar relationship to the Altai genome. This does not mean every population encountered a completely different species. The boundaries are not that clear. Ancient human groups split, drifted apart, met again, exchanged genes and split again. They were not clean branches on a diagram. They were rivers dividing and rejoining across deep time. And Denisovans may not have been the only hidden people in this landscape. Some genetic patterns hint that Denisovans themselves may have inherited DNA from an even older, still unidentified archaic population. A population that split from the ancestors of modern humans, Neanderthals and Denisovans, far earlier than the Neanderthal-Denisovan split. Researchers sometimes call this a super-archaic contribution. The phrase sounds dramatic, but it simply means ancient ancestry from a lineage we have not sampled yet. Could it have come from Homo erectus, which lived in Asia for more than 1 million years? Possibly, but that remains unproven. Homo erectus is an obvious candidate because it was widespread across Asia and survived there for an astonishingly long time. Yet without DNA, the connection cannot be demonstrated. There may have been other archaic groups we have never named, populations known only through a few unusual fossils or populations whose remains have not survived at all. Modern humans may also have mixed with groups that were neither classic Neanderthals nor classic Denisovans. The genetic evidence can sometimes reveal the outline of such encounters, but it cannot always provide a face, a language, a homeland, or a name. That is the unsettling beauty of ghost populations. They remind us that absence from the fossil record is not evidence of absence from history. Every new genome has the power to change the map. The first Neanderthal genome changed our understanding of modern human origins. The Denisovan finger bone changed it again. Denisova 11, the daughter of a Neanderthal mother and Denisovan father, proved that these lineages did not just occasionally brush past one another. They formed families. Then came ancient DNA from Tibet, Southeast Asia, and other regions. Each discovery added another thread. Each thread made the old picture less simple. What once looked like a world of separate human species is becoming a world of overlapping populations. Long migrations, repeated meetings, and ancestries flowing in several directions at once. And somewhere in the warm forests and southern islands of Asia, perhaps beneath cave sediments no one has sampled yet, lie the remains of people whose genes are still alive in the bodies of their descendants. We do not know what they call themselves, we do not know what they looked like, but their genetic footprints are still there, waiting for the rest of the story to be found. And that brings us to one of the biggest problems in the entire Denisovan story. Maybe the missing people are not missing because they were rare. Maybe they are missing because the Earth is very good at erasing bones. Fossilization is not the normal ending for a living thing. It is an extraordinary accident. Most bodies disappear quickly. Scavengers scatter them. Bacteria break down soft tissue. Rain carries away the smallest fragments. Roots push through bone. Soil acids dissolve it. In warm, wet environments, the process can be devastatingly fast. For a bone to survive tens of thousands of years, let alone hundreds of thousands, many things have to go right. It needs to be buried relatively quickly. It needs protection from animals, weather, and moving water. The surrounding soil must not be too acidic. Temperatures need to remain favourable. And even if the bone itself survives, the DNA inside it faces a separate battle. DNA is a fragile molecule. After death, it begins to break apart. Water helps chemical reactions that cut it into smaller and smaller pieces. Heat accelerates the damage. Microbes consume what remains. Over time, a complete genome becomes a pile of tiny damaged fragments, and eventually even those disappear. This is why Denisova Cave was such an astonishing stroke of luck. The cave lies in the Altai Mountains of southern Siberia, where cold conditions helped preserve genetic material that would have vanished almost anywhere else. The finger bone of a young girl, a few teeth, and scraps of bone were enough to reveal an entire branch of human history. But the cave may have preserved something unusual, not something typical. Think about the map of Denisovan ancestry in living people. Some of the strongest signals appear far from Siberia, in the peoples of New Guinea, Australia, the Philippines, and parts of island Southeast Asia. Other clues point toward Eastern Asia, the Tibetan Plateau, and perhaps southern China. These are not mostly cold, dry landscapes. Many are tropical or subtropical regions, places of intense heat, heavy rainfall, thick vegetation, and active soils. Beautiful places for life, terrible places for preserving ancient remains. A Denisovan skeleton buried in a Siberian cave might retain microscopic traces of DNA for tens of thousands of years. A skeleton buried in a tropical forest may leave almost nothing. Bone can become soft, porous, and chemically altered. Then it can dissolve completely. No skull, no tooth, no finger bone waiting in a museum drawer, just soil. And if the DNA is gone as well, the people who lived there can become almost invisible. This creates a dangerous illusion. When archaeologists find Denisovan remains in Siberia or Tibet, it is tempting to imagine those places as the centre of Denisovan life. But perhaps they are simply the places where Denisovan evidence had the best chance of surviving. It is like looking at the night sky through clouds. The stars you can see are not necessarily the only stars that exist. They are the ones visible through the gaps. The tropical regions may have been full of ancient human populations. Denisovans, early modern humans, Neanderthal-related groups moving through Western Asia, and perhaps other lineages we have not yet recognized. But warm climates have turned much of their physical history into silence. Still, that silence is no longer complete. Scientists are finding new ways to search for people even when their bones are gone. Caves remain especially important, not because every cave preserves fossils perfectly, but because caves can protect sediments from wind, rain, sunlight, and constant disturbance. Layer after layer of soil can accumulate on a cave floor, trapping tiny fragments of bone, charcoal, stone tools, pollen, animal hair, and sometimes traces of ancient human DNA. This sediment DNA is one of the most exciting developments in archaeology. Instead of extracting genetic material from a recognizable skull or tooth, researchers can take a small sample of soil and ask a remarkable question. Did any ancient humans leave DNA here? Sometimes the answer is yes. At Denisova Cave, DNA recovered from sediments has shown that different human groups occupied the cave at different times, including Neanderthals and Denisovans. In other sites, sediment DNA has identified ancient humans even when no human bones could be confidently recognized. It is a little like finding fingerprints in an empty room. But there is an important caution. DNA in soil does not always stay exactly where it was first deposited. Water can move particles through sediments. Burrowing animals can disturb layers. Later activity can mix material from different periods. A genetic trace may tell us that a Denisovan was present in or near a cave, but it may not tell us precisely who that person was, what they looked like, whether they lived there for generations, or whether they briefly passed through. And contamination is always a concern. Modern human DNA is everywhere. Archaeologists, museum workers, and even visitors can leave traces behind. Ancient DNA laboratories use strict procedures to separate genuine ancient molecules from recent contamination, looking for the characteristic chemical damage that accumulates over deep time. Even then, conclusions have to be tested carefully. A few fragments of DNA can transform the story, but they can also be easy to overinterpret. Proteins offer another possible path. DNA usually disappears first. Proteins, especially those locked inside tooth enamel, can survive much longer under conditions that destroy genetic material. By studying ancient proteins, researchers may be able to determine whether an isolated tooth or bone belonged to a modern human, a Neanderthal, a Denisovan-related population, or something more distant. This approach helped reveal that the ancient people associated with the Jahe mandible on the Tibetan Plateau were closely related to Denisovans, even though researchers could not recover a full ancient genome from the jaw. It is not the same as reading DNA. Proteins carry less information. They cannot give us a complete family tree or identify every episode of interbreeding. But in regions where DNA has been erased, they may provide a bridge across the gap. Improved dating methods are changing the search as well. The question is not only what is this fossil, it is also when exactly was it here. Modern techniques can date cave sediments, mineral deposits, and tiny traces of radioactive decay with increasing precision. That matters because Denisovan history unfolded across enormous spans of time. A cave occupied 50,000 years ago tells a different story from one occupied 200,000 years ago. Without reliable dates, bones, tools, and DNA fragments can be placed in the wrong chapter. Slowly, the map is becoming more detailed, not complete, not even close. But every sediment sample, every protein sequence, every newly dated tooth gives researchers another chance to find people who have been hidden by climate and time. So when a region contains no Denisovan fossil, the honest answer is not. Denisovans were never there. The honest answer is, we do not know yet. They may have been absent. They may have passed through briefly. They may have lived there for thousands of years. They may have mixed with modern human ancestors there, leaving descendants, but no bones. The evidence has to decide, and in many parts of Asia, the evidence has barely begun to be collected. The Denisovans are not only a mystery because they were ancient, they are a mystery because much of their world was built in places where history decays. And this is where the Denisovan story becomes most honest. Not where the answers are clear, but where the questions get bigger. We know Denisovans existed. We know they were closely related to Neanderthals. We know they interbred with modern humans, and that some of their genes still survive in living people. We have a finger bone, a few teeth, fragments of bone, a jaw from the Tibetan Plateau, traces of DNA in cave sediments, and a genetic legacy spread across Asia and Oceania. That is already extraordinary. But if you asked a group of scientists to draw a Denisovan face, the room would become very quiet. What did they actually look like? The known teeth are unusually large, with features that seem more archaic than those of Neanderthals or modern humans. The Jahe jaw is massive and robust, built for powerful chewing. Some genetic research has suggested possible clues about pigmentation, body form, and other traits. But these are still fragments of a much larger picture. We do not have a Denisovan skull. We do not have a complete Denisovan skeleton. We do not know whether a Denisovan walking through an ancient valley would have looked immediately distinct to us, or whether they might have seemed, at first glance, like another kind of human. Perhaps different Denisovan populations looked very different from one another. A group adapted to cold Siberian forests may not have resembled a group living in humid southern China or on the islands of Southeast Asia. And that brings us to another unanswered question. How many Denisovan populations were there? The name Denisovan can sound like it describes one people, one appearance, one way of life. But the genetics suggest something much more complicated. Modern populations carry more than one kind of Denisovan ancestry. Some ancestry components are especially associated with people in Eastern Asia. Others are found at much higher levels in people from New Guinea, Australia, and island Southeast Asia. These genetic signals do not appear to come from a single encounter with one small uniform population. They point toward a long history of separation, movement, and mixing. Maybe there were northern Denisovans in the Altai Mountains, adapted to cold continental climates. Maybe other groups occupied the Tibetan Plateau, surviving in thin air and intense seasonal conditions. Maybe still others ranged through forests, river valleys and coastlines farther south and east. It is possible that populations separated by thousands of miles had been evolving independently for so long that they were as different from each other as distant human populations can be, while still belonging to the same broad Denisovan branch. Or perhaps the word Denisovan is too simple for the reality. Perhaps it is a label we have placed over several ancient populations whose relationships we are only beginning to understand. Then there is the question of their lives. What language did they speak? The simple answer is that we do not know. Language does not preserve in caves. No Denisovan voice was recorded, no writing survives. No story was passed down with a name we can recognize. We can infer that they communicated, all humans do, and the movements, interbreeding, survival strategies and possible long-distance connections seen in the archaeological and genetic record would have required social learning of some kind. But did they have languages as flexible and expressive as those of modern humans? Did they tell stories? Did they sing? Did they name mountains, rivers, animals, and ancestors? Did parents teach children through words, gestures, imitation, or all three? We do not know. The same uncertainty surrounds their culture. At Denisova Cave, archaeologists have found stone tools, ornaments, and other remarkable objects. But caves can be shared spaces. Neanderthals, Denisovans, and modern humans all used the cave at different times. Sometimes the sediment layers are disturbed. Sometimes dates overlap. Finding an object in a Denisovan-associated layer does not always prove that a Denisovan made it. They may have made sophisticated ornaments. They may have exchanged objects with Neanderthals or modern humans. They may have used tools made mostly from wood, fibre, bamboo, leather, and other materials that disappeared long ago. The archaeological record is not a complete inventory of ancient life. It is a collection of whatever managed to survive. And perhaps the deepest question is what happened to them. Why did Denisovans disappear as separate populations? They may not have vanished in one dramatic event. There may have been no final battle, no single catastrophe, no last Denisovan standing alone in a cave. More likely, different populations followed different paths. Some may have died out when climates changed and habitats shifted. Some may have been absorbed into expanding modern human populations through interbreeding. Some may have been small and scattered already, vulnerable to the same dangers that threaten every isolated population: disease, low birth rates, competition, and simple bad luck. Their bodies disappeared, but not all of them disappeared genetically. A person living on the Tibetan Plateau today may carry a Denisovan-derived version of a gene that helps the body cope with low oxygen. People in Oceania and parts of Asia carry other Denisovan fragments, each one a tiny inheritance from meetings that happened tens of thousands of years ago. In that sense, Denisovans did not simply end, they became part of other people. So where will the next discovery come from? Maybe from a new cave in southern China, Laos, Indonesia, or the Philippines. Maybe from a museum collection, where an old tooth has been labelled as unknown for decades. Maybe from proteins preserved inside enamel. Maybe from a teaspoon of cave sediment. Or maybe from living genomes, as researchers find genetic patterns that reveal another ancient population hiding behind the Denisovan signal. That is the strange beauty of this story. The gaps are not failures of science. They are invitations. Every unanswered question tells us that the human family tree is still growing. And somewhere, perhaps in a cave floor, a forgotten drawer, or the DNA of a living person, another Denisovan fragment may be waiting to change the story again. And maybe that is the only honest way to end the Denisovan story. Not with a complete skeleton, not with a final portrait, not with the comforting feeling that the mystery has been solved, but with fragments, a small finger bone found in a Siberian cave, a few enormous teeth, pieces of bone that at first could have belonged to almost anyone, a massive jaw discovered high on the Tibetan Plateau, tiny traces of ancient DNA preserved in cave sediments long after the people themselves were gone, and, perhaps most remarkably, living human genomes carrying small inherited pieces of Denisovan ancestry across Asia and Oceania. That is all it took to change the human family tree. For most of modern science, human evolution was imagined as a fairly simple sequence. One species replaced another. One line moved forward while another ended. There were ancestors, descendants, and dead ends. The picture was never completely simple, but it often looked like a tree with clean branches, Denisovans helped break that picture apart. They revealed that our past was not only a story of replacement, it was also a story of meetings, of populations moving into the same valleys, using the same caves, following the same herds, crossing the same mountain passes, of people who had been separated for hundreds of thousands of years encountering one another again, sometimes competing, sometimes living beside each other, and sometimes having children together. Modern humans met Neanderthals. Modern humans met Denisovans. Neanderthals met Denisovans too. At least one young girl, Denisova 11, had a Neanderthal mother and a Denisovan father. Her tiny bone fragment gave us something almost impossible to imagine before ancient DNA. Not a vague sign of contact, not a statistical hint. A direct family connection between two populations we once pictured as completely separate. And she was almost certainly not unique. The genetics of living people suggests that Denisovan ancestry entered modern human populations more than once. Some of those encounters may have happened in mainland Asia. Others may have taken place farther south, perhaps among populations moving through island Southeast Asia and towards Sahul, the ancient connected landmass of Australia and New Guinea. The exact routes remain uncertain. The number of Denisovan populations remains uncertain. Even the boundaries of the name Denisovan remain uncertain. But the larger pattern is clear. Human history was never a neat march of one perfect species replacing all the others. It was a braided river. Groups separated. Groups returned. Genes moved across boundaries. Populations disappeared as distinct communities. But parts of them continued in the descendants of those encounters. A Denisovan person may be gone as an individual. Their village may be gone. Their language, stories, names, songs, tools, and traditions may be gone too. But a small part of their genetic inheritance can still be present in someone breathing thin air on the Tibetan Plateau, or living among the islands of the Pacific, or tracing family roots through Asia. That is a strange kind of survival, not survival as a nation or a tribe remembered by name. Survival as a fragment, a useful gene, a stretch of DNA. A biological memory passed quietly from parent to child for thousands of generations. And if that much can be learned from so little, imagine what may still be waiting. Asia is vast, its caves are numerous, its tropical forests are difficult places for bones to survive. Rising seas swallowed ancient coastlines where people may have lived for tens of thousands of years. Many fossils discovered long ago were classified before anyone could test them for ancient proteins or DNA. Many more are probably still underground. Somewhere in southern China, perhaps, there may be a tooth from a Denisovan population no one has recognized yet. Somewhere in Indonesia, the Philippines, Laos, or the high valleys of Central Asia, there may be bones from relatives even more distant than the Denisovans we know. Or maybe the next discovery will not be a bone at all. Maybe it will be sediment from a cave floor, maybe a protein locked inside ancient enamel. Maybe an unexpected pattern in the genome of living people, revealing another lost population whose body has not yet been found. The Denisovan story reminds us that the human past is not finished just because it is ancient. It is still being uncovered, one fragment at a time. If you enjoyed this journey into the mystery of the Denisovans, please like the video, comment and subscribe for more quiet history stories. And let me know in the comments where you're listening from tonight. The best way to support the channel is to listen on Spotify through the link in the description.